So we have another lecture today on methods of growth estimation, direct macroscopic counts, electronic enumeration methods, Weibull counts, and CFU methods, membrane filter, and turbidimetric. In my last lectures, we talk about the some detail about this quantification method, but here we discuss these methods in great detail so that you can understand more about the methods like direct methods and indirect method, and also methods which are differentiate between the Weibull count and non-Weibull count. Now, as in my last lectures on quantification, we talk about the general overview about the quantifications and their roles and their applications in the area of microbiology, both in the industrial applications and then medical applications. Then we have application in the area of most innovative techniques which are being used for different products which are produced from the bacteria. So for all, the estimation is important. So why estimation is important, why growth estimation is important, is a big question which we discussed in my last lectures about the overview. Now we will discuss some more detailed method today, how these estimation can be precisely calculated and precisely implemented in the field of microbiology to provide the best possible application both in environment, food industry, and of course medical industry in medicine, clinical medicine. So let's talk about the methods of, these are the some point of contents where we discuss about the methods of growth estimations, direct macroscopic growth, electronic enumeration, Weibull counts, and the turbidimetric method. Now first talk we will discuss on the bacterial generation time. Now as you are aware about the bacterial generation time, in my earlier lectures we discuss about when we discuss about the growth, that how the generation time is calculated, what is the significance of generation time, and what is how generation time is correlated with the growth rate. Now growth rate is inversely proportional to the generation time. So this is the key point in the whole quantification or estimation method. Now if you understand the relation between generation time and growth rate, then the purpose is solved. It means now you can easily identify the microbial world on which you are working for their application. This is, I am talking in terms of very general overview. For example, you have E. coli, and the E. coli is a recombinant E. coli with the any protein or any growth factors which are involved in that, and this E. coli is being produced through some methods, and if you are not aware about what is the growth rate and what is the generation time, so it is very difficult for you to get this bacteria at particular quantitative level, quantifying this bacteria at particular level, so that it may or may not be used for the application. So if you able to quantify this bacterial growth with the background knowledge about the generation time and the growth curve and growth rate, which is inversely to generation time, then it is easy for you to pick that bacteria or that strain for the purpose of any environmental purpose, for clinical purpose, or for food industry. So bacterial generation time is 1 to 3 hours. It depends on the bacteria. In some of the bacteria, and some requires even more than 24 hours. Some require even more than 24 hours, or some even high. So what kind of a bacteria you are using, it depends on it depends on the type of bacteria that is being used. Now the number of bacteria, for example, double in each generation. For example, generation time 2 to the power 5. It means you have, or we call them 2 to the power n. N is a generation time. So you have this number of generation means 2 into 5. 2 to the power 5 is equal to 2 into 2 into 2 into 2 into 2. 1, 2, 3, 4. 1, 2, 3, 4, 5 is equal to 32. So 32 number of cells are produced, or cycles are produced, when the generation time is to the power n. So this is the number of exposed as a power to the power n. This is the number of cells, accordingly say 32. if you have a 2 to the power 4, it comes to be 16, right. So, this is how you can calculate the generation time, and at every cell division. So, this is the example of cell division where you are dividing the cells into 2. It means from this cell to this 2 cells, the time required is called the generation time. Is called the generation time. I think this is clear to you. Now, generation time is important to learn the growth rate. This is a good chart to understand the total log values to calculate, because generally it is a log 10 logarithm, because it is exponential growth, right. This is exponential growth. So, this exponential growth is calculated into the log value, right. See. So, this chart gives you the generation time, generation number, say 0 to 20, and this is the number of cell 1 to 1.1 lakh something, then you have 0 to 6. So, these values are basically important for calculating the number of cells present. Now, when you calculate the log 10 value of these number of cell, that will reflect you to total number of cells which are present in that, like this, and this will help you to calculate the clinical side, for example, number of cells present in an infection, or if you are working on the infection control. For example, if you are working on infection control model and you simply calculate the CFU. In the CFU, you say, for example, after a treatment with some medicine, some drug, or some method that you design, you look into whether the infection is controlled or not, then this number is important, whether the numbers are reducing or numbers are not reducing or increasing. If numbers is increasing, infection is not controlled. If numbers are reducing, infection is controlled. So, how to calculate this number, right, is calculated through log 10 number, right. So, this is the simple method of the calculation. Now, bacterial growth curve, we several times discussed about the lag phase, log phase, stationary phase, and death phase, right. So, in lag phase, you have the starting cells are preparing themselves for growth, increase in number. So, in lag phase, no numbers are increased, numbers are not increased, but in lag phase, the cells are very active. It's not like that the lag phase is inactive phase. Lag phase is not a dormant or inactive phase. Remember, sometimes people thought the lag phase is a dormant phase. No, lag phase is, say, this is a lag phase. Cells are not at all growing in 1 to 2 hours, but after that it starts growing. So, people thought that cells are not growing, so it is a dormant. No, it's not dormant. It is a totally active phase where the cells are preparing themselves for enzyme productions, protein productions, so that they can form a better growth platform for the cells to grow. So, but there is no almost negligible growth present in terms of like a increase in number. But the duration of the lag phase may be 1 hour to 2 hours. It depends on the type of cells and metabolic conditions, or also nutrient media and growth conditions. Now, this is a plot. Several times we already discussed about this is the time plot, and this is the log number of the bacterial cell, where you can see the lag phase of some time, then you have a log phase, stationary phase, and death phase. So, this we already have discussed in great detail. So, I am not going to talk again. If you have any question, you can ask sometime. Now, in the log phase again, it's a rapid cell growth, generation time is a constant with exponentially increasing, and then cells are metabolically very active, physiology is very fast. It preferred the phase for industrial production, enzyme productions, metabolite productions, growth hormones, or recombinant proteins productions, right. In a stationary phase, cells are exhausted and they start a depletion state, and cell growth get depleted because of the exhaustion of the nutrient media or the nutrient components. And death phase, the cells are dying because toxins are start producing after a certain time, then nutrients are not available, they all finished, exhausted by utilized by the cells, and so ultimately cells are dying. Now, what are the requirements for the microbial growth? So, you have a physical requirement, then you have chemical requirements. In physical requirements, you have a temperature, you have pH, pressure, right, then some humidity in certain cases like water content. And chemical requirements, you have a media, you have a carbon compounds, you have a phosphorus, oxygens, and you can have different nutrients which can provide the energy for the cells to grow. Now this is a perfect plot showing the bacterial growth rate at the different temperature. So first we have the saccharophile, right? Then we have a sacharotroph. So saccharophiles are growing on the temperature below 0, right? And the optimum temperatures come to be about 10 degrees centigrade or 8 to 10 degrees centigrade. Similarly, sacharotroph are further increase in temperature, then mesophiles comes where the optimum temperature is about 37 degrees centigrade, between 38 to 40 degrees centigrade, but it is exactly 37 degrees centigrade plus minus 1 or 2 is considered the best optimum temperature for the mesophiles, which are the pathogens also, right? Then thermophiles, which has a very, very high temperature, so it can grow even at 70 degrees centigrade, or it can go to even more than 70 degrees centigrade in case of hyperthermophile. So these are the different 4 or 5 categories of the cells, right? So if you are growing, although we have discussed this part in the physiology, but here we are talking in terms of like growth, how growth is controlled, how growth rate is being maintained through these parameters. So if you can, suppose if you are working on saccharophile, what kind of a conditions to be given for the best growth rate? So what is the question here is that what is the optimum growth? If you are able to provide the optimum growth condition, your growth rate is fast. If you are not able to provide the optimum growth rate condition, the growth rate is slow, right? So in order to maintain the optimum growth rate, you have to provide the best possible conditions in that. Again, this is the quite busy slides. I'm sorry for that, but I think this is effect of the oxygen on the growth in various type of bacteria. As you know, the bacteria or microbes are classified on the basis of utilization and not utilization of the oxygen. Some bacteria they grow in the presence of oxygen, we call aerobics. Some are non-anaerobics or facultative anaerobics, right? So aerobes and oxygen, obligatory aerobics bacteria which require the oxygen and which cannot grow in the absence of oxygen, and facultative anaerobes are the bacteria which can both aerobics and anaerobics conditions, and obligatory anaerobes are completely anaerobics where there is no oxygen is required, right? Similarly, aerotolerant anaerobes where we can have like tolerate the oxygen. So there are different categories on the presence because oxygen may be toxic for certain cell. They react oxidations and they cause oxidation and produce singlet oxygens and a lot of reactions, and they may lead to death of the cells. So this is another conditions where you have to look into while growth. So if you are not aware about the physiology, if you are not aware about the conditions, growth rate conditions, then for you it is difficult to quantify the growth at different parameters. So you have to provide the best possible parameters, optimum conditions for the growth rate. Now the method of growth estimation. What are the different method? Different method of growth estimations we have slightly discussed in an earlier slide. The CFU, that is the colony forming unit, we already discussed. This colony forming unit, that is this one, is the method where we call them as a direct method, because you grow the cells, count the colonies and count the number of cells corresponding to each colony, because single colony means originated from a single cell. Then you have a membrane filtration method, direct microscopic method. You can directly visualize the cells, how many number of cells you have under the microscope, that can be done. Then we have the electronic enumeration, or we call Coulter counter method, and most probable number, that is MPN method, which is also a method which directly you can count the coliforms and other bacteria. Now in case of indirect method, you have a turbidity method, that is the spectrophotometry or colorimeter method. Then we have a metabolic activity where you can have the growth at different level. You can get the whole mass, dry mass of the cells. Then dry weight also can be calculated. So let's talk about the direct microscopic count. In case of direct microscopic count, you have simple slides, and these slides have been taken. For example, this is the slide, and you have a So this is a kind of a chamber you make, and in this chamber you put the cells, and these cells are put on each chambers. Now what you can do, you can calculate the number of cells in each chamber, and after the each chambers is number of cells calculated multiplied by the factor of the volume in which these cells are present. For example, you can calculate this. For example, if you magnify this chamber like this, so length, breadth, and then you have a cover slip like this, like this. So this cover slip, which has a maybe height of few millimeter, for example 0.01 or 0.1 millimeter or 0.01 millimeter or 0.1 or 100 microliter, whatever, depend on the oil immersion that you are taking, right? For visualizing these cells, the cover slip make a length. So you multiply it with length, breadth and height of this cover slip, give you the volume. Now how many cells present in a one chamber will give the number of cells in one volume. So you accordingly you multiply it with the factor with the 1 ml to centimeter, from millimeter to centimeter, by multiplying by thousands, and if it is volume to the cube of it. So total number of cells can be calculated reverse back in the 1 cc or 1 ml. So that refer to the 1 cc refer to 1 ml. So this is how you can calculate the total number of the cells in that. This is the method where you cannot differentiate. The limitation of this method is that you cannot differentiate between the live and dead cells. So you may calculate the all the way dead cells as well as the live cells. How many dead cells we have? So that will tell you the growth curve where the for example here. So if you are calculating the cells in the exponential phase, in the exponential phase the growth is continuous and you don't have any dead cells. But when it reaches to the stationary phase or decline phase, then you may have a chance of the dead cells. So if you are taking the culture of exponential phase or log phase, you have a little chance of having dead cells, whereas if you are taking the culture at the 12 hour, 24 hours, reaching to the decline phase or stationary phase, then you may have a chance of the kind of a dead cell also. Now this is a grid which is made, and there are 5 steps have been defined, where on this slides you have a microscopic cells are present. So this is the visual presentation what we have just explained, and you can count in each chambers and multiply it with the factor. So this is how the calculation is being done. You can go through this calculation. 1 upon this 2,50,000 of milliliter. Now this can be reverse calculated to the 1 milliliter, 1 ml. How many cells present in 1 ml? So this is the reverse calculation, but it gives you the direct. Now it is very, very difficult for you to calculate the total number of cell in a whole slide or in a whole stream, because there are millions of cells are present. So it is always better that you make a grid, and in the grid you can have number of cells easily count in one grid. So this is a simple method of calculation of the cell through the microscopic method, or we call the visualization method. So tools, what is this called? It's called Petrov-Hauser counting chamber. It is a special slide, just like this we have shown earlier. And this Petrov-Hauser counting chamber is basically the method where you can, so this is the Petrov-Hauser chamber, counting chamber. In this chamber you can calculate the cells. Now then come to the Coulter counter. The Coulter counter is something, I cannot make good drawing, but I think it is a kind of a machine where you can have a cell and it is connected with some circuit, right, plus minus, and the machine is just like a I can give you the analogy of this machine is like a note counting machine in the bank, right? You just put the notes and it give you the, all the notes can be counted easily through this machine, 1, 2, 3 with the sensor. Similarly, when the one bacteria come here in this orifice, there is a sensor present which sense the number of cells coming out of it. So every cell which come out of it will be sensed and give a deflection or in the 1, 2, 3, 4, 5 like that in number 1, 2, 3, 4, 5, 6. So digital estimation of the cell can be done through this Coulter counter method. And the Coulter counter method is again, the limitation of Coulter counter method is again that it cannot differentiate between the live and dead cells. So, live and dead cells is not at all possible with that. Then what are the advantages of these two methods? So, these are the direct method, these are the indirect method where you can have the direct method where you can directly count the cells, but the limitation that the dead and live cells both are counted. Motile bacteria are difficult to count. For example, Proteus mirabilis is one of the example where it is very difficult to count because it's moving, it little motile. So, when you put on the counter method, for example, if you put here number of cell like this in this counter, say for example, out of 10 cells, 1 cells moved out and reached to another counter, another chamber, another cells move another chamber. So, those cells which move, right, motility, high motility, they cannot be counted with this. So, they count, but not very precisely. So, this is one of the disadvantage, whereas it is very rapid method, very fast method and easy to handle, no extra skills are required, right. They are very useful for speed and essential for mill quality testings and clinical laboratories, etc. So, the limitation with limitation, these two methods are having very good high quality counting, and it has advantage that is very efficient, very fast, but with the limitation of the non-differentiating between the live and dead cell. So, if you want to calculate the dead and live cell together, or if you want to calculate the live cell only, then for that the microscopic method are present. So, differentiating live and dead cell, there is a called the simple confocal microscopy. Now, this confocal microscopy give you the difference between the live and dead cells. Now, similarly, FACS, flow cytometry, right? Flow cytometry also give you the clear-cut difference between dead and live cells. For that, you need to have a dye. With the dye is differentiating between live and dead. Dye is I can give you the name of the dye like a cytonine dye and then PI dye. So, cytonine dye is binding to the cells, intact cell surface, and PI dye bind to the DNA. So, if PI dye concentration which give you the red color is more, it means more cells are dead, right? And if cytonine dye is more, that is green color, then it means the live cells are more. Then you can merge these two images together in the microscope and you can find the variation between the live and dead cells. You can compare the live and dead cells together. So, whichever is more is more. For example, if red color is more, it means the dead cells are more. If green color is more, it means the green color live cells are more. So, these are some methods which can be easily quantify properly the cells in this method. Now, this is all about these two methods. Now, the other methods we will discuss in my next lecture soon.